All right, welcome everybody to today's Microbial Genomics and KBase webinar, where we're going to talk about doing gene feature analysis in KBase. My name is Benjamin Allen. I'm here at Oak Ridge National Laboratory, and I'm an outreach coordinator for the KBase program. So what we're talking about on today's webinar is continuing the theme from the last webinar, which is doing sort of the basic processes and protocols for microbial genomics using the tools that we have on KBase. So the last time we discussed this, we looked at drafting isolate genomes. And so you can see in the tutorials section of the narratives browser, the narratives navigator, that we've got a link to this drafting isolate genomes narrative. And oops, that's the bottom of it, but you can see all of the information from that tutorial and webinar and this narrative. But we're looking at this this gene feature analysis one today. And so what we did in the last one was that we generated a assembled and annotated isolate genome, and we're going to now sort of take that as the basis for some further investigation, where we want to know more information about what the genetic content and character of this organism is. So, so to do that, we're going to go through a couple of series of steps, but the overall goal is to identify and isolate the features that we're interested in. We're going to build a multiple sequence alignment in KBase, and then use that multiple sequence alignment as the basis of constructing and searching for hidden Markov models in our target genome. And then we're just going to look at some of the apps that we can use to further analyze any given feature. So the organism that we're looking at is this Sulfobaculum zeaminens. That was it's a delta proteobacteria living in mangrove soil kind of sediment. So and it's associated with some respiration processes for heavy metals. And so what we're going to do is what we're going to try to look at today is specifically try to see if we can understand if this organism has any capacity for mercury methylation, so methylating environmental mercury and integrating it into its overall metabolic profile of this organism. So this is we're looking at some previous research that was done by my colleagues here at Oak Ridge, which is in this aptly titled "The Genetic Basis for Bacterial Mercury Methylation" paper that came out in Science about a decade ago. And as the title would have you understand, they did indeed establish the genetic basis for mercury methylation. And they did that through a lot of experimental work. But what this has allowed us to do is sort of take this finding that they have, that there's this 2-gene cluster of HGCA and HGCB that form the complex required for mercury methylation in bacterial metabolism. So what we want to do is use KBase to search for these features against our target organism. So the way that we're going to do this is that we're going to replicate some of what these researchers did. And so this picture here, of course, shows the loci of these gene clusters in 6 different organisms here that are all delta proteobacteria. So what we're going to do now is use KBase's public data, or what I should say we've already done, but we use KBase's public database to add these genomes from this paper. So this little GIF here is showing you how to do that. I'll just kind of show you in real time here, where you can see there's this public tab here, and that has all of the NCBI RefSeq microbial genomes in there. So all 234 some odd thousand genomes. And so what we did is we added these 6 genomes for the same reference genomes that this study used here. And so here you can see I've got all of these genomes. And, for instance, in this Geobacter metalloreducens here, if I go back up to that top chart, I can see there are these aliases for these terms here. So if I look up GMet 1240, I believe is what it said there. Yes, so I can then see the genes that are mapped out here from the original paper. So these are the features. This is just sort of showing this is how you can search for these features in the genome object to indeed verify that the aliases are there, etc. But what we want to do is take all of these features listed here in this diagram, and we want to build what's called a feature set from them. So that's taking all of the gene features from these genomes and building a set of them that we're going to use for the downstream processing. So what do we do to do that? We build feature sets. So there is an app in the utilities section called Build Feature Set from Genome. Right now, the way this works is that you have to build out the feature set from each individual genome and then merge them into a single feature set. So you can kind of see here, what I do here is for each one of these genomes, I selected the genome, selected the target gene, and then I built a feature set with this kind of name here, where I just appended the name of the gene to the end of the name of the organism. And sure enough, what that builds is a feature set that is just a single feature that I pulled from this organism. If I click this feature ID here, it's going to open up another tab. It's going to have information about the feature, like the sequence and this kind of information. If you're looking for that, that's one way you can look at it. But overall, what we do is we build a feature set for each one of these genomes corresponding to those in the paper. Okay, sorry, just looking at the chat here. It looks like most people can see my narrative pretty well. Maybe I'll do a little boost of the resolution there so you can see better. So what we did is we then took all of these feature sets of the HGCA gene and okay, maximize the window. I'll stretch it out, yes, just a little further here. I don't always do full size because you try to get it as close to the proper resolution for YouTube as possible. But this should work fine. Thanks for letting me know in the chat there. So again, we built out these feature sets, we merged them, I gave them a description. These are the genes from this particular paper, and I gave them this name of HGCA. Actually, that's the wrong one here below. I'm going to delete it and pull out the correct one here. So here we go. This is the merged feature set with all of the genes from these organisms from this Parks paper. So I do this for both the HGCA and HGCB features in these genomes. So yeah, I'm sorry that you're having trouble, Ahmed. If no one else is having trouble, I'm just going to keep going. But yeah, I hope you're able to get it resolved. And if other people are having like these issues, just let me know so we can pause. But anyway, so we're building this out for HGCB as well. So we do the exact same process and build out this HGCB as well from all of these genomes using the build feature set from genome tool. You can, of course, when you're building these feature sets, you can select any number of gene feature IDs for the protein coding genes within your genome. but in this case, this is kind of a way you can just isolate something for a single feature and then merge it into this larger feature set. So that's what we've got here. We've now got two feature sets, HGCA and HGCB, and that's what we're going to use for the next processes here, which is building and trimming multiple sequence alignments. So this is another graphic from this Sparks paper, where you can see the what's marked with these little green arrows here are these organisms that we've selected here. But more importantly, what they're showing, this is sort of their characterization of a large number of genomes. And basically what they've identified is the specific amino acid sequence within the protein that are affiliated with the functional capacity of these two genes to methylate mercury. And so this cluster here is in the HGC a gene where you've got this cap helix here with this highly conserved region. And then there's these transfer features that are in the HGCB cluster that deal with shuttling between iron and sulfur. So this is related. These are the two essential genes for performing this mercury methylation function. So what we want to do is establish if our new organism that we've just sequenced and we've just assembled and annotated and built the genome from, we want to see if it has these two genes. And this is going to be sort of the signifier that we've got to hit, is that it's got these highly conserved regions. So to do that, we want to recreate what they did in this paper with a multiple sequence alignment. And so what we do is use this muscle tool, which is this is a kind of canonical computational biology tool. I'm sure many of you have built multiple sequence alignments before, but if you have not, it's very easy to do in KBase. Similarly, we put in our feature set that we built, give it a description and an output name, and then we run the app. If you've never run apps in KBase, I assume most of you have, because this is more of an advanced webinar, but you add the app from this app panel, and then you fill out the parameters, and then this reset button usually shows a run button until you run it. So that's how you run things in KBase, is that you fill out the parameters and you click run on the app. And this, there's really not that much configuration you have to do with this tool, especially if you're building something from a known set of data. But this is what you get out of it. Is it's kind of hard to see in the window here, but you get this clustral alignment map here, where you can see there's annotations for conserved, highly conserved, semi-conserved, and non-conserved sections of this this genome. It's kind of hard to see, like I said, but at the bottom you can download the clustral format file, where you can look at it in a text file or whatever kind of text editor you prefer to do these things in. So yeah, let me see if I can't really. Yeah, it's kind of hard to see in this. Maybe if I actually shorten it, there we go. There you can kind of see it a little better, where it divides the two sides there. We can actually see the parts that are aligned here. And sure enough, we actually can see here this conserved region for the cap helix section here, this section right here. So this indicates that, indeed we've been able to kind of rebuild the MSA as they had it in the paper, and these things are lining up. The next thing that we want to do, so we do the same thing once again for the HGCB, and you can see there's these same kind of highly conserved regions are also present across these organisms that we use for the reference here. What we do after we build these multiple sequence alignments is that we want to trim them. So these are pretty closely aligned in length, and there's a lot more variability in some of these other ones. So we actually want to trim this down to trim off some of this extraneous non-conserved amino acid sequences in our multiple sequence alignment. So we do that with this another kind of canonical tool, G-blocks. So you build your multiple sequence alignment with muscle, and then you can trim the ends of it with G-blocks. Very similarly, you pick the multiple sequence alignment file, you give it a description, and you can give it an output name. Again, most of, depending on, how closely related and how much, homology there is between these, all of the feature sets you have, you may have to trim more or less and set some of these parameters, but because we're dealing with something known here, we don't have to do too much experimentation in the parameters with these. These are, which is to say, you can usually trust these with the default settings to get you something useful. So now what we've done is we have built and then trimmed our 2 multiple sequence alignments for each of these genes in this gene cluster. And so we want to use these MSAs as the basis for a search. So we have these strings, and we want to search for them in our target genome. But more importantly, we are not the literal strings, but more so the patterns of the amino acid chemistry here are then used to build a hidden Markov model for this search. So to do that, we use this tool, Hammer, again, another popular tool. They've got their own, most of these tools you can find on lots of other websites, but the reason to use it in KBase is that you can reuse all of the data that you've put in and share it and do this kind of stuff that we like to promote within KBase. So we have our target genome here. This is the one that we built in the last tutorial narrative. And then we want to run the Hammer search against it. So we take that trimmed multiple sequence alignment, we set the target. This doesn't necessarily have to just be a single genome. You can use another feature set. You can use a whole set of genomes, so you can potentially search against hundreds of data objects at the same time, or even if you have like an annotated metagenome assembly, and you're trying to do some profiling of your metagenome, you can also use the Hammer search tool for that as well. So again, usually the default parameters are pretty straightforward with these as far as finding a hit goes, but this is going to tell us if there are any genes in our target gene related to this multiple sequence alignment. And ta-da, sure enough, we have a highly aligned gene. This is a very good score here. So that this is, more or less is a direct hit. So this is exciting. So we can see that this disulfobacillum zeaminens does indeed have this HGCA cluster. They were, I'm not sure why this says 109% for what it's worth. That was actually a difference between the last version and the current version of Hammer. So there may be some weird scoring there, but yeah, nevertheless, this is a fully aligned between our inputs MSA and what we searched for in our target genome. So we've got a hit, and sure enough, the output of this is also a new feature set that contains this hit. So I'll just click that here. And this is what it built out. The output of this Hammer search is a new feature set containing this protein in our newly assembled genome. So that's kind of hard to look at here, but you can see this conserved region here. That was the highly conserved region that was established in the previous paper. But it's also just generally pretty close to the overall length that we had in our multiple sequence alignment that we used for the search. Similarly, we do the same thing with B, and this is maybe a little more interesting to look at. You can see the white background one here is another hit, very close in the overall alignment length, very low E value. So that indicates that these are likely the same gene. But you got a lot of other hits too. So this is kind of an interesting thing with this organism. You can see it's got all kinds of oxidoreductase or heavy metal reducing or ion translocating complexes here. So it's doing a lot of moving a lot of electrons around between different kinds of heavy metals or other environmental molecules here. So but most of these are not hits. There may be some aligned regions here you can see, so that might be other points of investigation. But again, we're mostly looking for this one specific function, and it's calling it ion translocating oxidoreductase complex subunit B. That is means that even though whatever this it was annotated using PROCA in our previous tutorial, that's just the term that's been assigned to this, and so it doesn't necessarily describe exactly what it does in this kind of specific mercury methylating context, but that would kind of indicate that the biochemical function that's been theorized is pretty close to what we're looking for, some sort of metal reducing and methylating function. So that's good. That's 2 hits. So again, I could build out a list of hundreds of delta proteobacteria, right? When they did this initial paper in 2013, right? I think this is like 70 something or 60 maybe organisms that they used. I could, if I wanted to spend the time, I could build out this whole genome set. I could grab all of the new delta proteobacteria that have been sequenced since 2013 and just run the, you can use this finding now. This is kind of the exciting thing about KBase is that, we've built a data object that represents this key finding from this paper that then I can just use to search against, whatever organism, whatever metagenome or, that I get, and that will help me understand, if there's any mercury methylating potential within those organisms. So you can do this with any number of gene features, or this is kind of nice because it's just 2 gene features. It's not a huge, multi-complex, kind of set of genes, and there are very strongly conserved regions in there associated with the functionality. But this is just kind of what's cool, I think, about how things work in KBase is that we're really building a data object that represents the finding of this paper. So you can imagine kind of doing this for all sorts of previous findings and using them for future search or future analytical purposes. And this is very fast to do. I mean, running this whole process takes maybe an hour. These are very fast tools overall. So, so I've got those 2 hits. That's exciting. I can use that for future analysis. If I want to continue to use the MSAs or the feature sets in there. So what else can I do with these feature sets or multiple sequence alignments or hidden Markov model searches in KBase? There's a couple of exciting things. One thing I always like to show people is paper blast. This, as you might think, you can take the protein sequence from anything. So this is the sequence I pulled out from this new organism. I just took the protein translation and dropped it into paper blast here. And this search is, I think, against PubMed mostly. that uses, a homology search to tell me something about the other papers that are out there that have, sequences similar to this search term. So, again, if I wanted to build out more sets of interesting organisms, I could use this to kind of see what's out in the literature. One of the things to kind of connect this to the future of KBase that I think is important is, we're building this thing that we call knowledge engine right now. As I've sort of shown you, we've got all of these public reference genomes in the system. What we're trying to do is build out some new features and functionality that will allow me to search across, use hammer searches from these custom MSAs to search across all 230 some thousand genomes of bacteria that we have in the system. So we can't do that yet, but that's kind of look back at us maybe in maybe 2 years, and I think we'll have some of the first versions of this loaded on the system where you can just do these kind of custom searches against all of the reference data in the system, and build that out in your narratives, or as we add new genomes or sequence, you can rerun these searches and continually manifest that data into objects in the KBase system. We think that this will be very much like a future direction of the science here, where people might be curating whole custom sets of features and this kind of stuff to use for characterization and other things. There's a whole nother narrative about what I'm going to show you all. It's called Build a Gene Tree. It's this last tutorial down here in the tutorial section. It has a little more detail about doing this, but just as an example of what we can do, where I want to then do some sort of similarity and dissimilarity assessment of this new gene that I have against all of these other genes. So just doing this with a small set here, usually when you are building these gene trees, you might use potentially hundreds of different organisms. But KBase has a lot of things that you kind of you run the same apps over and over to kind of build new stuff in the previous data sets. So the previous app that we showed, merge feature sets, we use this one again to take the HGCA set that we built initially, and then integrate this new gene into from our resequenced Disulfobaculum zeaminens here into the larger feature set. So I just called it, HGCA DX and gave it the date that I ran this. And so that just says, okay, this is the original data set with this new organism in it. And then I kind of rerun the same process. I rebuild the multiple sequence alignment. And you can see that it's added this one in here. And I can kind of see how it aligns and see where there's conservation or semi-conservation. But moving forward, we kind of do the same thing here where we trim it. Then we use this tool, build phylogenetic tree from MSA using FastTree. This is just a way you can build a tree that can show you the dissimilarity between whatever gene you're putting in, or between all of the same genes and across this set of organisms. So what I can see here is that this one is a little unique. It's not clustering with some of these other ones neatly, but you can see its degree of dissimilarity between some of these 4 in the bottom 2 branches versus these Geobacter sulfuridenses, some of these canonical delta proteobacteria. So that's nice. I can build out these trees. I can do the same thing with HGCB here, where I build out this tree, and very similar as far as the dissimilarity between these genes across these organisms. So So there we have now built, so we've constructed MSAs from our set of features. We've trimmed those. We built, aligned multiple sequence alignments across all these organisms, and we've used that multiple sequence alignment for the basis of a hammer search. And now we are building out a gene tree showing the similarity and difference between these genes across these organism set. So that's kind of the end of the proper tutorial here. I want to show just a couple of other things before I get into some of the questions, if there's any questions that are there. Just want to show you a couple of cool things you can do in KBase. So we built out this new feature set here. What I'm going to do is click these three dots underneath or right next to the name, and you see I get these buttons that come out. This first button here says show apps with this as input. So I click that. What that does is creates a filtered set of tools that can take feature set as input here. So you can just see there's some other tools, mostly in the sequence analysis and comparative genomics section that you can find here. And just in particular, just want to show some of these bottom ones here. These might be interesting to you, especially if you're looking at environmental microbiology or microbial ecology. These are some existing tools with curated sets of search terms, right, they're pulled from existing data. You can see, if you look at the info section, there's information about what some of those, where these search terms come from. But if you're, say, looking for hits for certain types of metabolic activity, like maybe I want to search for metals, and I can see, okay, there's iron and manganese oxidation, so it's searching for these specific kinds of gene features. I can do this for all sorts of numbers of things. What I'm actually going to put in is not the feature set. I am going to what I could do is build out a genome set with all of these. I don't actually have that genome set, but what I'll do is just select a genome here. So this is that same disulfobaculum one that we generated in the first narrative, and I'll just run this these metals and nitriles. Not actually, let me do these selenium here. That's maybe more exciting. So now I can search for let's give it a name here. Search for these specific element, or specific metabolism for these elements within this organism here. So I run that, and now it's launching. Similar, if you're looking for carbohydrate active enzymes, right? KZ families. This ends up being a pretty common search. You can similarly do search for this. Do some custom searches here with these curated sets of hammer profiles. So I'll just do the top one here, do a couple here, but similarly give it a name. And I don't know if you're seeing what I'm doing here with naming. I recommend doing this always, where I will append the date of the run to the end of the output file data name, because sometimes we update apps, or sometimes I ran something and then I realized I needed to rerun it. This just means I can kind of iterate using the same app parameters without having to reload the app and refit all of the parameters. I might just rerun it and change the date at the end. So I'm not sure how long this will. Oh, there you go. Right as when I asked is when it came up. Very good. So yeah, now this search with HMMs of elemental bioelement families came up, and there's hits here. You can see, I'm going to just open this report in a separate window, like this tab says, and interesting. It looks like it went ahead and ran all of the terms, even though I set a fixed set of terms, but nevertheless, it gives me an overview here of hits for these specific elemental metabolism hammer profiles that it searched for. So the gray ones here mean it's not present, and the white ones mean that it has a hit. So you can see under nitrogen, right, it's just got this nitrogenase iron protein here, but none of these other ones are present in the heat map here. I think if I hover, yeah, it'll tell me how many hits, and it will give me the names of the hits, the gene terms for these specific hits. If I want to, look into the genome and build it out, I can just see, right, there's a couple of different hits across these bioelement search here. Let's see, did the other one finish? It's still running there. That's exciting, and I think this is okay. This also does not generate a. Okay, sorry. I think I actually have explained the parameters wrong. This set's not what it searches for, but it's the parameters set the feature sets that come out of it. So I'm assuming, yeah, okay, so that's right. There's no hits here for the metals that we searched for. So it didn't build out that feature set, it looks like. I don't think, yeah, it would be at the top of the data panel here. So this doesn't set what it searches for, it just sets which feature set you want to build. And that's useful because maybe you don't want, combined feature sets for all of these different elemental search terms. But you can still kind of do some profiling here of the genetic capacity of this organism using this specific search tool. Let's see, did the KZ one? Okay, yeah. So similarly with KZ, you get a very similar kind of output here. But yeah, it did not generate any features set. So those are some examples of some of the things you can do with hammer searches on the system. If you want to do some phylogenetic marker searches as well, you can do that if you're trying to do more taxonomic work. But I'm going to just delete this one for now. So okay, yeah, I think that's mostly all I wanted to show y'all today. Let's see if there's any other tips or tricks I can show you here before I get into the question. I'll just say, most of these apps, or almost all these apps we show today, are in the sequence analysis section. There's different ways you can search for, search for genes or other features in your data. There's, classic stuff like BLAST, or FAMA is another tool that was recently added that's pretty interesting as far as doing some functional profiling based off of some curated sets of data. So, but we're actually going to kind of focus more on feature profiling in the next Microbial Genomics webinar, where we're going to talk about domain annotation, building functional profiles. And that's all. I'll show more of this stuff in the next webinar. But if you ever are trying to see, okay, maybe there's some apps that are not fully released, but maybe there's some cool stuff, you can always go into the beta apps. And if you ever want to help us test any beta apps, please sign up in this beta testers pool here in the Q&A document. We'd love to get some of these out, but we need lots of testing and feedback. But similarly, I can say I want to see what kind of tools take an MSA. I can see if there's any new stuff. It doesn't look like it, but yeah, just another kind of KBase tip there. If you're ever trying to see if there's any experimental apps you can run, you can always check out the beta apps there as well. So
thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.